rise as we receive the Vice Chancellor's procession. Vice Chancellor's procession, directors of academic programs, directors of institutes, deans of faculties, students, Provost College of Sciences, Provost College of Postgraduate Studies, the Inaugural Lecturer, Principal Officers of the University, the Vice Chancellor's procession. Standing, we'll take the first and second stanza of the national anthem, followed by the university anthem. The national anthem. greatest universe. Please, you may be seated. Our very highly cerebral and distinguished Vice Chancellor of our great university, Professor Edoba Omorege, Senior Advocate of Nigeria, Your Excellencies, top government functionaries, Your Royal Highnesses, former Vice Chancellors of our university and heads of institutions, other principals of our great principal officers of our great university, the provost, deans, and directors, the inaugural lecturer and family, professors emeriti, other esteemed professors and erudite scholars here present, my Lord spiritual, greatest university staff and students, invited guests, gentlemen of the press. We welcome you heartily to this 335th in the inaugural lecture series of the University of Benin, to be delivered by none other than a distinguished scholar and professor of pathogenic microbiology, Professor Mrs. ONAIWU IDAHOSA ENABULELE. On the topic, minute prokaryotic serial assaulters of man on the prowl, be on your guard. May I now invite the registrar, Mr. Ademola Bobola, to introduce the Vice Chancellor and the Vice Chancellor's procession. The registrar, sir.
Mr. Vice Chancellor, I want to seek your permission to continue to abide by the protocol as duly established by the representative of the university's PRO, Mr. Emmanuel Oreka. Mr. Vice Chancellor, I have a task before us today to start off this program, and that's to present your own worthy self and other very eminent scholars and drivers of the administration of this university at this particular time. And it is my sitting university librarian, who is the custodian of our books and publications, Walter Soyo Medicine, Esquire. I move to the other side of the divide here, and we have quite a very loaded entourage of the Vice Chancellor on that other side. First, the Provost of the College of Postgraduate Studies, the Vice Chancellor will allow me to make this remark. At the meeting of council recently, that college was added to the array of academic segments of this university, and we have coming up to that position, filling up the position for the first time, the provost of that college, Professor David Izeko. And welcoming him, because he has always been the only provost. But thank God today,
Inaugural Lecture Series of the University of Benin. Today's lecture is the 7th in the Department of Microbiology, the 46th in the Faculty of Life Sciences, and 6th to be delivered in my tenure as the Vice Chancellor of this great university. I'm pleased to inform you that the new and old students are resumed for the 2024 and 2025 academic session, and lectures have begun in all faculties, schools, and institutes. I therefore wish to sincerely appreciate all staff and students for their enormous sacrifices that ensured the successful completion of the 2023-2024 academic session. Distinguished ladies and gentlemen, it is my pleasure to introduce our lecturer for today. She is Professor Mrs. ONAIWU IDAHOSA ENABULELE. title of our lecture is Minute Prokaryotic Cereal, Assaulters of Man on the Prowl. Be on your guard. First of Mrs. ONAIWU IDAHOSA ENABULELE was born on the 25th July 1955 in Benin City. So the family of Mr. IDAHOSA IDAHOSA and Mrs. IDAHOSA Igbe of Yoloke and Igbe family of Benin City. She had a primary education at Matthews Primary School, Benin City. She had the first certificate in 1967. She then attended the African Girls Grammar School, Benin City, where she obtained the West African School certificates in excellent grade of grade 1, 1972. That was the highest at that time. Thereafter, she gained admission into the University of Benin, where she obtained the Bachelor of Science degree in microbiology, graduating with a first class degree. Please, another round of applause. This was in 1979. In 1978, she undertook the mandatory one-year National Youth Service program at the Institute of Continuing Education, IC, Benin City, from 1978 to 1979. In her quest for more knowledge, Professor Mrs. ENABULELE obtained the following additional degrees: the Master of Science degree in Microbiology in 1984 from the prestigious Ahmadu-Bello University, Zaria, and then a doctorate degree in pathogenic microbiology with specialty medical bacteriology from the University of Benin. Professor ENABULELE joined the services of the University of Benin in 1979 as a graduate assistant in the Department of Microbiology and rose through the rank to the position of professor in the year 2009. She has held several administrative positions within the university, such as member of the Central Entrance Examination Committee, member University of Benin Student Certificate Screening Committee, head Department of Microbiology, chairman Task Force on BENN Enterprises, member BENN Committee on Business Ventures, and of course, culminating excellent service to the university as Dean, Faculty of Life Science, between the year 2014 and 2018. Professor ENABULELE belongs to several professional bodies and associations, such as member Nigerian Society of Microbiology, member Nigerian Environmental Science Society, the member of the Learning Societies. She is a recipient of the following awards: Federal Government Secondary School Male Scholarship, 1970 to 1972, Principal's Prize, African Girls Grammar School, Benin City, 1971 to 1972, the Benin Area Joint Board Undergraduate Scholarship Awards from 1974 to 1975, the Federal Government Undergraduate Scholarship Awards from 1975 to 1978, and the Dean's Prize, Faculty of Science of the University of Benin, 1977 to 1978. Professor ENABULELE has successfully supervised and co-supervised 3 MP and 30 master's programs. She has also supervised 14 PhD theses and very numerous undergraduate projects. She has published extensively and currently has over 70 publications in several reputable local, national, and international journals. Clearly, she's a very distinguished scholar. I'm sure you all agree. Professor ENABULELE was an external reviewer to the Nigeria Journal of Botany, African Journal of Biotechnology, and Human Journal of Science. She has served as an external examiner, assessor, resource person to some Nigerian universities as a member of several Nigerian Universities Commission UC accreditation teams to several universities in Nigeria. She has also attended several local, national, and international symposia, workshops, and conferences. Professor Ms. ENABULELE hobbies are reading and singing hymns. She's a devout Christian of the Church of Nigeria, Anglican Communion denomination, where she's a parish lay leader. As well as patron of the African Youth Fellowship. She is also a Knight of St. Mary, Kesem, the African Diocese of Benin. Professor ENABULELE was married to the late Dr. Richard Osaramabo ENABULELE, the SY Medical Director of St. Margaret Hospital, Benin City, and the union is blessed with many children and grandchildren. Distinguished ladies and gentlemen, it is my pleasure to invite Professor Mrs. ONAIWU IDAHOSA ENABULELE to deliver a lecture.
The Vice Chancellor, sir. Deputy Vice Chancellor's Administration, Academic, Campus, a distinguished registrar, acting bursar, acting librarian, other principals, officers of the university, professors emerita, deans and directors, my highly esteemed professors and other academic staff, my highly esteemed university staff, students of Great Uniben, my distinguished guests, ladies and gentlemen, I'm delighted standing before you now to present my inaugural lecture, which is the 335th in the series in the University of Benin, the 46th in the Faculty of Life Sciences, the 7th in the Department of Microbiology, and the 1st in my area of specialty, medical bacteriology. My lecture today is on some seemingly inconsequential, minute organisms. They appear inconsequential, but they are small and mighty, in the sense that they can attack the high and mighty in the comfort of their secure palaces, as well as the humble, lowly, and poor. And it is titled, Minute Prokaryotic Serial Assaulters of Man. Be on your guard. I wish to thank the Vice Chancellor for giving me the opportunity to present this inaugural lecture. And I hope that at the end of it all, we will all have an idea of what these organisms are, what they do, and how to protect oneself against them. That is, how to be on the alert and be on your guard. I'm glad to have you all here. You are welcome. Introduction. Prokaryotic organisms are single-celled organisms that differ from other cells that make up higher animals and human beings and plants, in the sense that they are nuclear, like a membrane, and they do not have organelles like mitochondria. There are two groups, the bacteria and archaea. The archaea reside in very, very extreme environments, like hot springs and thermal springs, hydrothermal vents. But the bacteria are with us in the usual environment. And the majority of them are beneficial to mankind in the sense that they help in the sustenance of life, in that they are major players in sustaining the biochemical cycles of life that enable life to go on Earth. Besides, they are used in the production of a number of foods. But a few have developed attributes that enable them to assault important structures and functions in man. And once they do that, they cause harm, disease, and sometimes death. In doing this, they cause a lot of pain, worry, fear, anxiety, financial, emotional stresses to man. They need to provide some information on what these organisms are and how to protect oneself against them informed this lecture. Such information will help in individual and community-oriented strategies to prevent and minimize their assault. Now, what are these organisms like? The bacteria are typically very small, ranging in size from about 0.2 to 0.4 micrometers in diameter. That is about 1/12,000th part of an inch, if you know what an inch is. 1/12,000th of an inch. That's why you cannot see them with your naked eye. You can only see them when magnified. And when magnified under the microscope, individual cells appear with various shapes, depending on the species. Some are rod-shaped, as you can see there. Others are spirals. Others, like the one that causes vibrio cholera, are comma-shaped. And yet others are spherical, like you have there in figure 1. And to be able to see them at all with the naked eye, you have to grow them. You have to put them on special nutrients where they can grow. That's the only way you see them with the naked eye, and that's what you do when you are about to identify them and check for antibiotics to which they are susceptible. And when you culture them, what you see are what you have in figure 2. I have there, this is the very popular E. coli. You can see the color. Forming colonies. Colonies are clumps of cells, millions of cells coming together to form clumps. And then that's the popular Staph aureus that we hear about. But to get their detailed structure, the detailed structure of the prokaryotic cell, what you have on the board is what it is like. That's the detailed structure of the bacterial cell, with a single chromosome at the center, no nuclear membrane, no organelles, but ribosomes in the cytoplasm, which is surrounded by a cell membrane, and then a cell wall, which is made up of a unique substance that is unique to bacteria, peptidoglycan. Some have external to this cell wall a capsule, but not all. The bacteria that can move about, the motile ones, move by means of flagella. Those are thread-like structures extending from their surfaces. Some have one, like the one you have in this figure, others have many. And then to attach to cells, to human cells, and to inanimate objects like surfaces, they have pillar. To be able to grow them, they require nutrients. And the particular ones we are interested in today are those minute, those few that are able to harm man and cause disease, known as the pathogenic bacteria. They are pathology producers. That's why they are named pathogenic bacteria. And to grow them, they require nutrients and appropriate environmental conditions. For pathogenic bacteria, what they require are organic nutrients like sugars and proteins and fats, just like man. We say they are chemoheterotrophic. There are others that do not use organic nutrients. Environmental requirements, they require temperature ranges of 25 to 45 degrees centigrade. They are referred to as mesophiles. There are others that grow in the fridge and freezers. Those are cycrophiles. There are others that grow in high temperature environments. Those are thermophiles and hypothermophiles. Pathogenic bacteria are neutrophiles. That is, they grow at neutral pH. And some of them require oxygen, like man, for growth. They are the aerobes. Others can grow only in the absence of oxygen. And yet others grow both in the presence and absence of oxygen. These are the facultative anaerobes. The human body is optimal for all of these conditions that I have mentioned. Made up of organic stuff, 37 degrees centigrade, neutral. And so once these bacteria get in there, they are able to grow. And as they grow, metabolize and grow and increase, they release products of metabolism that harm the human cell and can result in disease. That's the assault we are talking about. Bacterial diseases have been with man since history, and it is unending as we speak. We have been able to get a solution, a permanent solution to the onslaught of bacterial infections. Now, how do they do this? That's what I refer to here as the assault. Mr. Vice Chancellor, sir, for a bacterium to be able to assault, it will have a reservoir, as shown in table 1 before you. The reservoirs are many. Man himself is a reservoir because we harbor them inside and outside our bodies. Animals, the environment, birds, these are reservoirs, as shown. And from these reservoirs, these organisms are kept on the prowl. They are moved about by various processes such as air. Those who have meningococcal meningitis, for instance, you just breathe it in from the air. From the human carrier, he coughs, he speaks, and then you breathe. And you come down with meningitis, and so on. Other carriers or vectors include food and water. Those who have cholera, you drink that water that is contaminated, you are down with cholera, and so on. And once they get deposited at the appropriate portal in the body, then they begin to express their weapons of attack. The first set of weapons they express are the defensive weapons. That is, the weapons they use to defend themselves against the body's reaction. Because the body itself, every human being has a defense system that protects it from onslaught by foreigners, including bacteria and other organisms. So what these organisms, these pathogenic organisms do is once they get deposited, they begin to elaborate products that will attack the defense mechanism of the man. And there are many different types of products depending on the bacterium. And once they succeed in overcoming these defense mechanisms, they now begin to elaborate their offensive weapons. These are the weapons with which they attack the human cell and function. And these weapons are of two types. They are those they use for invasion, the invasive weapons, either through direct penetration of the entire skin or through production of several invasins. What are invasins? Shown in table 2 there. Invasins are enzymes and proteins that destroy body tissues. Once these invasins are produced, they begin to dissolve the body tissues. And by so doing, once the body tissues are destroyed, of course the function is gone, and there is a problem. The other way is to produce toxins. And there are two types of toxins. There are the endotoxins that are natural parts of the gram-negative bacterial cell, a group of bacterial cells, produced by all gram-negative bacteria. And these are produced while the organisms are either alive or dead in the human body because they are part of the cell wall. When the organisms are alive, they are produced in form of small bits released into the cell to attack the cell and cause massive inflammation. And what is the result of inflammation? When there's inflammation, the results are pain, increased temperature, redness, and cellular dysfunction. And that's what causes this disease one experiences. The other group of toxins are the exotoxins. These are released by different types of bacteria, as shown in table 3 there. And again, there are different types. There are the neurotoxins that affect the nerves, the nervous system, including the brain. For example, the organism that causes tetanus produces tetanospasmin. What tetanospasmin does is to act on the nerves and cause it to be continuously contracting without stop. And that is tetanus. Once there's continuous contraction, the muscles of the throat, the lungs or whatever contract, then there's a problem. That's tetanus. Another group of exotoxins are the ones that cause diarrhea, like the one produced by the cholera organism. What they do is, as they grow in the intestines, they cause fluid to be drawn out of the tissues. That's why the person gets dehydrated into the intestine, and once there's much fluid in the intestine, then the person begins to pass a lot of feces. That's cholera. So there are different types depending on the bacterial species. So by these ways, these organisms cause diseases. We can't go into all. Details are in the booklet. And as a microbiologist, someone in the field of microbiology, what do? The role of the microbiologist is to conduct research and surveillance studies, producing data for health practitioners to guide their identification, to guide their detection, to guide the trends of the disease in the population, and to eliminate the population, the bacteria from the population and the environment. And of course, to manipulate this bacteria for the use of man. And I have been doing just that, teaching that and researching on that in a number of infections. But here I cannot tell it all. I have chosen to speak on 3 very common infections, what I have done. And the number 1 is wound infections. At the time I was doing my PhD here, wound infections was very common. In fact, a colleague, one thing that drew me, a very senior colleague was in UBTH here for many months after having a cesarean section. And I asked, what is this? Why won't this thing go? Let me see. Besides, I lost 2 close relations to diabetic wounds. So I said, let me see, what are the organisms that causes wound infections? And so I went ahead, infected wounds, traumatic wounds. And then later, in the days of kerosene explosions, when there were so many kerosene explosions, sending shivers down the spines of everybody, I said, let me see, what are these organisms? And then, of course, diabetic foot ulcers. And what I found are what you see in table 4. E. coli, P. aeruginosa, Klebsiella, Enterococcus, fecalis, and others, is basically the same in all of them. Publications are as shown there, and this works. Basically the same set of organisms. Mr. Vice Chancellor, sir, what strikes one about this is that all of these organisms that you see here are normal inhabitants of the human body. They are there in our bodies, either on the skin or inside our bodies, although some can be isolated from the soil and water. So what that means is that these infections, most of them came from your own body, the body of the patients. For operation wounds, either from the body of the patient or from the surgical instruments, or even from the bodies of the surgeons and the environs and the theater. And so, to guard against, to be on your guard against these infections, as doctors as surgeons, they know that they have to keep bacteria off themselves, off the theater, and off the patient before they start. We don't have to dwell upon that. But as individuals, because we now know that they are part of our own body, it's good to have a very clean shower. It's good to make sure that we treat any infection in any other part of the body. We have to make sure that our blood sugar is okay. And then after the operations, we have to be very careful. We shouldn't be playing with the surgical site. We should not allow the surgical site to get wet, because this will drag organisms into those sites. These are ways to protect ourselves. And for traumatic wounds, we cannot say we do not have traumatic wounds. The infections are usually inflicted by the objects by which the wounds were inflicted. And what to do, traumatic wounds, is to wash under running tap water and then rinse with saline and then put a dressing. You are sure you are done. It's not going to be infected. And for diabetics, I want to counsel, please don't hide your wounds. Do not, as many do, because it could lead to amputation, it could lead to death when it goes into the bloodstream and cause generalized sepsis. So diabetics, never hide your wounds. Once you get wounded, visit the health specialist immediately. And please, for diabetics, try not to walk barefooted, because that's the problem. We hope, Mr. Vice Chancellor, sir, the second set of infections we looked at are infections of the digestive tract, that is the gastrointestinal tract, like diarrhea, cholera, typhoid fever, dysentery. These are all infections of the digestive tract caused by a good number of organisms. Many cases are not reported. Many of us have diarrhea and we don't bother to go to the doctor. They are not reported. But the burden is high, because World Health Organization had reported in 2024 here that there are over 1.7 billion cases of children's diarrhea. That tells you that the burden is high. We are not talking of adults worldwide. So what we were doing in the lab really is to look at sources of infection. Where do these infections come from? As a young mother who lived in the heart of Benin City, Upper Sakuba Road, my own children had a fair share of diarrhea. And there were so many pet animals around, dogs and cats in almost every home, including this domestic chicken, not the farm one, everywhere. And my children crawling on the floor. They had their fair share of diarrhea. And I just thought, could these things be carriers of some of these organisms? And I decided to look at them. Look for organisms that are called childhood diarrhea in pets, household pets. And what did we find? We found that 34%, 15% of household chicken carry the enteropathogenic E. coli. That is the strain of E. coli that causes diarrhea in over 80% of children. And dogs and goats, 10%. And then the E. coli that causes traveler's diarrhea and adult diarrhea too are the enterotoxigenic E. coli ETEC. We also screened, and we found that cows and cats were culprits. There are the publications there, ENABULELE and ENABULELE. And how do we get this infection? Consumption of food and drinks, directly or indirectly, contaminated with the fecal samples of these animals. Mr. Vice Chancellor, sir, we also looked at vegetables that are normally eaten raw. Cabbage, carrots, lettuce, cucumber. In fact, we made about 50 samples of salad by ourselves, washed, and we decided to screen. What did we find? The salad we made, 24.3% of them had salmonella. That's the one I want to talk about. Other diarrheagenic agents were there. But the salad we made, just washing like we normally do, out of that 50, 24.3% carried salmonella alone. We did not stop there. Fortunately, much later, I had a student from the north. And at the time he came, there were epidemics of diarrhea and all of that. So I asked him, you go collect vegetables from the farms where they are made in Kano State, where they are produced, right from the farms. And let's have them, let's screen. All this talk about diarrhea and cholera in Kano area, Katsina. Let's see what it is all about. And we screened. And what did we find? That's table 6 there. 20% of lettuce carrying salmonella, 48% of lettuce carrying Shigella. That is the organism that causes dysentery. Even the carrots we buy along the street there, 26% there carrying salmonella species, 24% carrying Shigella. And then Yersinia, another deadly strain of bacterium that causes diarrhea, very bad. Some of these vegetables were also carrying them. And what are the sources of these vegetables? Either, because they are right from the farm, either irrigation water, or the soil, or best droppings, and all of that. So, these vegetables, we need to be worried when consuming them raw. We are coming to that. And then, we also screen some sources of water supply for domestic uses in some rural areas in Edo State and Adamawa State. In Edo State, we screened rivers in Oriomon, local government area. River Rawado, flowing from Agbo, from Oligi, through Agbo, Urewa and all of that, so Wesi, in Oriomon, local government area, where this body of water serves as source of water supply for the inhabitants. We also went to Edo Northeast, Ogbofe River. We also screened for agents that cause diarrhea at points where they use this water solely for their domestic purposes. They don't have boreholes. And then we went to Adamawa State. That year, there were cries of so much, how many epidemics of cholera, outbreaks of cholera there. And I told a student whom I had, go there, let's check. They don't have boreholes because their water level is very low. So they depend on 3 main rivers. The whole of Adamawa North Senatorial Zone depend on 3 main rivers. For their, so in fact, I have photographs here where you see them pumping water into jerrycans for sale to the general public. And we screened. We also screened borehole water and well water in Orion, Abudu, Ihu, Ogan. We screened. These ones now have boreholes. And what did we find? All the samples we screened, coliform count higher than the normal of 0 per mil recommended by WHO. Coliform counts are used to estimate water quality because coliforms are in our tummies. We carry them in our intestines. So when you find coliforms in water above a certain level, you know that that water level has been contaminated with fecal matter. Being contaminated with fecal matter, that means there can be pathogens in them. So the coliform counts of all these water samples were higher than that recommended by WHO. And in some of them, we actually found typical enteropathogens like vibrio cholera, the rivers in Ogbofe and Adamawa State. We also found not only bacteria that cause diarrhea, also worms like Ascaris, Tracheris in these water samples. And some of them, when we screened them, had the relevant virulence factors as shown in table 8. The relevant assaults, weapons with which they cause diarrhea and dysentery and cholera. Which means this water, if you are fortunate to drink what has more than enough of the dosage, you come down with any of these infections. The rivers in Adamawa State in particular, all of them, high percentage, up to 50% of water samples from those rivers carried vibrio cholera. Any wonder why you hear of cholera outbreaks every year in northern states? And so, Mr. Vice Chancellor, sir, we screened these 3 infections. We also screened sexually transmitted infections. What we were after there was looking for risky factors. Because we all know that sexually transmitted infections are transmitted by direct contact from man. The source is man. you are either a carrier or you are a patient. And by direct contact, you get these infections. So all we were looking for here was looking for risk factors. And what risk factors did we find for sexually transmitted infections? As we all know, multiple partners. Secondly, dosing. The females here should take note. Dosing predisposes to sexually transmitted infections. Dosing, anal sex, oral sex. All of these are risky factors for sexually transmitted infections. In addition, for those of us females who use IUDs, if you look at the results we found here, in table 9 here, 53% of those who use intrauterine devices succumbed to infections, sexually transmitted infections, as compared to 19% of those that use other types of contraceptives. And the result is, in the process of fixing these things, you push in microorganisms, and besides, when you yourself are not so hygienic, you don't observe hygienic measures, organisms that are normally resident are pushed into your genitals, and you come down with sexually transmitted infections. Mr. Vice Chancellor, sir. So, to protect, to be on guard against digestive tract infections, for animals, you need to keep them away from areas where food is processed. You need to keep them away from areas where toddlers play, and you need to keep their living environment clean, and you need to take them to the veterinarian regularly for checks, so that even if they have these infections in them, they can clear them before they get transmitted to the household. And then for vegetables, washing is the key, and the triple wash is recommended. That triple wash, you wash very well under running clean water, then you wash with water with drops of vinegar, then you rinse. That's the triple wash. Once you do that, you are sure to get rid of all the pathogenic organisms. They won't survive vinegar. You are good to go. And in addition, please, for our women in the kitchen, prevent vegetables from getting in contact with raw surfaces on which raw meat has been processed. Because many of these animals bear these agents. And when you cut raw meat with your cutting board and you don't wash properly and you begin to cut with your vegetables, particularly those you eat raw, you are likely to transmit infections from them to the household. So ways to be on guard. Wash the vegetables. Make sure they don't come in contact with raw surfaces. And for water, for those who use such water, who do not have even boreholes, we are told the coliform counts are high. And for boreholes, the way they get contaminated is from leachates. Leachates are the refuse dumps around. Water will leach and leach pathogens into pipes that may be broken or rusted, and your borehole water gets contaminated. The same thing with septic tanks. And so it is advisable when you are constructing your septic tank, you keep it at least 30 meters away from your borehole. That way you are sure that leeches will not come in from there to contaminate your water bodies. And for sexually transmitted infections, we have said multiple partners. All of us know that. And women who dote, many of us are very fond of that. We put perfume in the water, or we put a small disinfectant in the water and begin to use it to wash. That is doting. You will remove the normal flora that protects against infection and allow pathogens to grow. So women, stop doting. These are ways to be on guard. Mr. Vice Chancellor, sir, one very bad sexually transmitted agent is Chlamydia trachomatis that is reputed to be responsible for infertility in couples. That that it damages the female tube and they cannot have babies. But infertility in our own work, we did not find any association with females, but we found in males, in the sense that chlamydia trachomatis affected sperm count, affected the sperm vitality, affected the sperm structure. In over 200 males, we screened those who have chlamydia trachomatis. So we counsel, please, our males should also come up when we are investigating infertility. It may not always be a female problem. I'm not saying it's a male problem, but sometimes it can be a problem of the male. So let's not move back. Let's be there for ourselves. Mr. Vice Chancellor, sir, in all of these infections, all of them, every one of them, as if you look at what you have on the screen there, all of these wound infections, respiratory tract infections, urinary tract, ear infections, and all of that, all the wound infections, everything we isolated, all the organisms we isolated were multiple antibiotic resistant. That is, they were resistant to more than 3 classes of antibiotics. More worrisome is the fact that some of them contain genes that produce an enzyme, extended spectrum beta lactamase, that destroys different antibiotics, ENABULELE enzymes. They destroy antibiotics like ampicillin, augmentin, cefixime, ciprofloxacin, septrin, streptomycin, gentamicin. One organism carrying a gene will produce an enzyme that will destroy all of these antibiotics. So what can we use to treat such an infection? Because of this, the WHO has cried that we are approaching antibiotic era, pre-antibiotic era. So we should be careful and mind our environmental footprints. Mind what we do. Don't take antibiotics without prescription. When you take complete dosage, don't prescribe for others. Don't use leftover. All of these we select for resistant strains. And one approach to this now is development of new drugs, especially from plants. And we have also been into plants, looking at those that can destroy bacteria. And in these, we have screened a number of plants, including the common scent leaf which we use, akata leaf in Edo, that is Rawophia, grape peels. We have screened many. And good enough, all of these, we are very active against bacteria. Even garlic mixed with some bacteria, they were all very active. As you see in that plate, the bacteria there, these ones are the ones unexposed to garlic and lactobacilli. These ones are the exposed. You can see the cells are fewer here. And besides, some cells are already undergoing lysis. Which means these herbs are very effective, but we must be cautious because in one of them, akata leaves, because each time we do this, we also do toxicity studies. In one of them, we found that akata leaf in Benin, that's Rawophia vomitoria, damaged the kidneys. The kidney parenchyma became very, very grisly, and the glomerula that is responsible for urine filtration became very, very small. So we must be cautious, and we are advocating more research into all of these medicinal herbs, so that not just research into the antimicrobial activity, but also into the toxicity studies. And recommendations generally for the government is that, and people, community, is that governments and health agencies should educate and empower communities on team approaches to preventing and controlling infectious diseases. Team approach. Don't desecrate your water bodies. Be hygiene conscious and all of that. And there is need for collaboration of research efforts by universities, research institutes, hospitals, and the National Center for Disease Control, so that information gotten from research can be disseminated, and then they can be used to plan strategies for prevention and control. Then the antibiotic problem, the One Health solution. Like I said earlier, we must be mindful of our environmental footprints, what we do to the environment, because these antibiotics, they are pushed into the environment by effluents from hospitals, from animal husbandry, where they use antibiotics to treat animals for growth. And they all go into the soil. And from there, they are leached into the bodies of water, and then back to man, and back to the soil. So we must be very careful. All of these agencies, what we refer to as the One Health approach, must work together to stem the tide of antibiotic resistance. In conclusion, Mr. Vice Chancellor, sir, that these organisms are there everywhere is not in doubt, and they are kept on the prowl, kept moving by various means, such as air and even the food and drinks we take, is not in doubt. And the assault is unending, because these organisms, they are adapting every day to resistance to available drugs and getting more deadly weapons of assault. So, Mr. Vice Chancellor, sir, as we have highlighted possible sources of these agents, we should be wary to be on guard against these agents. And we advocate rational use of antimicrobials. Acknowledgements. Mr. Vice Chancellor, sir, I give thanks to Almighty God, who has kept me alive and in health, and given me all the good things in life. All. He has made me fruitful in all my undertakings. When you know my story, you will know that God has been so good. You heard my name. You heard what my name is. Glory be to God Almighty. ONAIWU is a story. So we thank God. I owe my late parents a debt of gratitude. Thanks to my late ever-caring husband, he was a dependable asset to me. I'm very grateful to my mother's siblings. Some of them are here. The Igbe family. They took on the role of parents when, in my teenage, I became orphaned. They didn't let me suffer. I enjoyed everything a child will enjoy. Till today, my cousins, they are still so supportive. So I'm grateful to you all. I appreciate my wonderful children and grandchildren. My children, they didn't disappoint me. At a point, I almost gave up my career for the home front. They did not disappoint me. I appreciate my former vice chancellors, Professor Adamu Baike, who employed me, Professor Oji Oshodu, and Kuba Injo of blessed memories, who facilitated and announced my promotions after a long delay. I appreciate Professor F.F.O. Owumesi for his support when I was going through trying times. He knows what I'm talking about. He's here. Thank you, sir. I appreciate all my colleagues and workers in the Department of Microbiology for their wonderful support. I appreciate the huge contributions of my former students, MSc and PhD students, such as Professor Ofori, Professor Musa Dairu, Professor Ekase, Professor Akinjagunla, Musa Samuel Azebeni, Brenda Isiche, and many others. Kennedy Nibadin, Nifre Nibadin. These are all Ewosa, Professor Ewosa Yamu. These are all my former students whose work, most of their work I have used in this lecture. I want to appreciate my family friends, my husband's friends. Professor Okere, Professor Igieba, Professor and Mrs. Ofodu, Professor and Mrs. Ima Okudi. They have remained friends indeed, because even after I left my husband, even after my husband left, they have not left me at all. They are still very much with me. I want to appreciate all my teachers, from secondary, Mrs. Ibuje Secondary School to University, Professor Gbemi, Professor Sani is here, Professor Elkundayo of blessed memory, and many others. I thank you all, my colleagues in microbiology, my colleagues in physical sciences, and other departments, other faculties in the university. I appreciate you all. You have all been wonderful. I do not want to leave out the administrative tasks, because in their own way, they have all been very good. I have here Mrs. Ifeadini. I have a Giato here. I have a Takbo here. There are very many. There are administrative staff that have played major roles in helping me. The technical staff, Mrs. Oragawu and others, Mr. Kabina, they are all here. I appreciate them. I want to appreciate especially Professor B. Alinde. I thank you so much. We've done some work together and he's been wonderful. I thank all former deans of life sciences. I want to thank you all for coming. You have all made my day. Thank you so much.
A resounding ovation for our distinguished lecturer. We can do better than that for her. Thank you so much, man. Just to make it simple. That was quite a robust and educative lecture. I believe a lot of us learned so many things tonight. It's my singular honor tonight to invite the Vice Chancellor, Professor Deba Norege, SAN, for a recap of this evening's lecture. The Vice Chancellor, sir.
Distinguished ladies and gentlemen, I'm sure you will agree that you have had a huge taste of brilliance, haven't you? Yes. Just remarking to our dear sister that our lecture reminds me of the wonderful outing of Professor Musahai Irafora himself, here present. Emeritus Professor, the small and the minute was at the party. Something of that nature. That was the title of his inaugural lecture, and it's wonderful to hear brilliant minds engaging. Distinguished ladies and gentlemen, what you have listened to is the brilliant lecture of Professor Mrs. ONAIWU, IDAHOSA, ENABULELE. Mine is to give a summary of what we have listened to. Prokaryotic organisms, otherwise known as bacteria, are unicellular organisms that lack a membrane, bionuclear, and organelles. Pardon me, I'm not a microbiologist, so I would make some missteps, but just pardon me. I'm trying to learn a couple of these terms from my wife, who I say was a biochemist, but I see in an infant stage at this, even as a professor myself. Pathogenic bacteria are able to absorb man's organ system either by invasion of the system or toxin production, causing infectious diseases, disability, and sometimes death, according to Professor ENABULELE. Obviously, it's not me, it's talking. Man, animals, and the environment serve as natural reservoirs for them, and through direct contact, air, food, water, insects, and animal vectors are transmitted to man. Factors such as poverty, natural disasters, floods, keeping domestic pets, trade and travels, etc., facilitate spread locally and internationally, keep them on prowl. Of course, you remember the COVID-19. I'm sure something close to this must have caused that very serious global pandemic coming from China. President ENABULELE's research contributions in this area have centered on documenting the causative agents of wound infections, infectious disease of the gastrointestinal and reproductive tract, as well as their susceptibility to antimicrobial agents, possible reservoirs, and risk factors. Plants such as Osimum cadicimum, scent leaves, that's the English name, Citrus paradisi, as grapefruit, peels, Philatus amarus, Uvaria cheme, and others were also screened for antimicrobial and immunomodulatory activities. Thank you for clapping for me. There are reagent agents such as different pathological types of E. coli, Salmonella, there is SPP, what does that mean? Vibrio, cholera, and Shigella, SPP, etc. were all detected in different sources of domestic water supplies. She went to Ogbese River in West Southwest, not far away from here, to make that discovery. And 3 other major rivers, such as which are domestic water sources in Adamawa State, and almost all of these isolates were resistant to many classes of antimicrobial agents in use. All investigated plant extracts elicited remarkable antibacterial activities against some clinical bacterial isolates. It is advocated that utmost care be taken when consuming these plant preparations, as evidence shows that some of them may be toxic to some organs. Staying safe and being on guard is via strict personal, household, community hygiene, sanitation, avoidance of risky behaviors, and instituting appropriate monitoring and management system for surface water bodies by government. Ladies and gentlemen, please can we put our hands together again for our dear sister, a first-class graduate of this university, somebody who has distinguished herself and was elected Dean of Faculty of Life Science of this university. We're very proud of her. Our dear own Professor Mrs. ONAIWU, IDAHOSA, ENABULELE. Thank you very much for your attention.
We now invite members of Professor ENABULELE's immediate family to join her on the platform as she's decorated. Members of our immediate family, only the children, please, only the children. We welcome Mr. Richard ENABULELE. We welcome Mr. Duwa ENABULELE, Mr. Lola ENABULELE, Mrs. Jennifer Obaseki, Barrister Mrs. Philippa ENABULELE, Mrs. Ajibola ENABULELE, Deacon Osayuki Obaseki, Dr. Amadi ENABULELE, Engineer and Mrs. Sobo Yejo, the Igbo family. Also, we welcome the grandchildren Jadon, Jeremy, Cheron ENABULELE, and Usoreme and Orobusa Obaseki. We'd like to welcome specially to this evening's lecture distinguished personalities and dignitaries who are here present in no particular order. We welcome Honorable Justice Peter Isiboro, Honorable Justice Ikwaroba, Honorable Mrs. Ighodaro, you're welcome. We welcome the former Vice Chancellor of this great university, Professor Faraday Oluwese. We welcome the former Vice Chancellor of the Ambrose Ali University Epoma, Professor Dennis and Lady Nike Agbonaho. We welcome Barrister Musahari Ogeva Okumbo. We also welcome tonight our former registrar of this great university, Mrs. Otasore Oshodi. We welcome the former Deputy Vice Chancellor Admin, Professor Jacob Eherobo. We welcome former Deputy Vice Chancellor Admin, Professor Mrs. Adesua Osaho. We welcome Professor Emeritus T.O.K. Audu, Professor Emeritus and Mrs. Raymond Elaho, Professor Emeritus Julius Ihatua, Professor Emeritus Felix Okene. We welcome Professor Emeritus Musahare and Professor Mrs. Egafuna. We welcome also Professor Bamidele Soni. We welcome Professor Joseph Oselehai. We welcome Professor Frederick Ekhaise. Professor Felicia Febu, you're welcome. Professor Sir Andrew Ubu, you're also welcome. Professor Solomon Omoniho, you're welcome. Professor Chris Okaka, you're welcome. Professor Eruke Egbabe, you're welcome. Professor Amowe Ovewe, you are most welcome. Professor Henrietta Obo, you are also welcome. We welcome also Professor Faith Akeneboso. We also welcome members of the clergy here present. We welcome representing the Anglican Bishop of the Benin Diocese, Venerable Osaze Uwadia. You're welcome, sir. We welcome from St. James Anglican Church, Venerable Saro Ibinosa. We welcome Venerable Moses Onowuke. We welcome Venerable O.A. Idoi. We welcome Venerable Sam Isen. We also welcome Reverend Canon Iyekebolo Enofe, Reverend Canon Samuel Ugiagwe, Venerable Professor Osawoi, you're welcome. We welcome Professor Mrs. Bosede Ajayi, Professor Sylvia Ogwana, you're welcome. We welcome Professor Mike Omoigberali, Orator, University of Benin. We welcome Professor Kajiagbe. We welcome Professor Faustina Idu. We welcome Professor Olajire Bosede Ajayi. We welcome every one of you to tonight's inaugural lecture. As we bring this lecture to a close, I'd like to crave your indulgence that you rise as we take the national anthem and the Union anthem. The third stanza of the national anthem and thereafter the University of Benin anthem. The second stanza, I beg your pardon. Okay, so we do Union anthem first and thereafter the national anthem. The second stanza. Second stanza of the national anthem. Thank you for coming. Please remain standing as we allow the Vice Chancellor procession exit the auditorium.